Welcome back to this next video in which we are discussing the human papillomavirus entry mechanism. Okay, so I just would like to make one final comment about the endocytic pathway, and then we'll move on to the second type of endocytosis, Caviola-mediated endocytosis. So, my comment on the endocytic pathway, the final thing that I wanted to say is, how do these little vesicles, such as the endocytic vesicles here in blue, or this vesicle here in green, or this vesicle here in orange, or this vesicle here in mint green, how do these actually get through the cytoplasm of the cell? Because you might be wondering, remember the cytoplasm of the cell, something that I took loads of time earlier on to stress. It's not just a gloop, it consists of loads of cytoskeletal components. There are fibres everywhere. How do these vesicles get in between those fibres? Well, the answer is they're small enough to go through the gaps between the cytoskeletal components. So that's the first thing I wanted to say, that they can get through the gaps between the cytoskeletal components. So there's no problem with them not being able to get around unless the cytoskeleton breaks down and lets them through. They can get through the gaps between cytoskeletal fibres. The second thing to say is that we think that actually the cytoskeletal fibres effectively function as little tracks for them to follow. So we think that there are lots of cytoskeletal fibres that will go between membranes of the cell. So for instance, there might be cytoskeletal fibres from the plasma membrane all the way to the early endosome, and the endocytic vesicles, they can follow these little cytoskeletal fibres that link the membranes that they want to go between, and we think that this is important for them being able to find their way around the cell. So I just wanted to make that little comment about how the endocytic vesicles interact with the cytoskeleton. Firstly, that it's not going to obstruct their passage, they can get between the fibres, and secondly, that actually they seem to even follow fibres along, and that seems to be crucial for how they get around the cell and find their way to the place they're meant to go. Okay, so there's the comment made about the endocytic pathway. Let's now talk about caveolin-mediated endocytosis. So firstly, I'm going to do the speech that I've done many times before, just to remind you. Remember, endocytosis of human papillomavirus is controversial. We do not know at present completely how this works. Lots of papers are present that have studied this, and they all seem to be pointing to different answers. It may be that different human papillomavirus types use different secondary receptors, and that those different secondary receptors use different endocytosis pathways, which would explain some of the discrepancies in the different papers. Alternatively, it might be that all the human papillomaviruses use the same secondary receptor, but that this secondary receptor has multiple different ways that it could be endocytosed. So those two possibilities are there. In this video, I'm going to tell you about all these three different types of endocytosis, and the horrible reality is that there are even more types of endocytosis than these three that are believed to be present within cells. They're not understood really at all, which is why I'm not going into those ones. However, let's now go on to caveolin-mediated endocytosis. So let's discuss how this is going to work. So the first thing, obviously, to talk about is caveolae. What are caveolae? So caveolae is the plural, and it literally means little caves. The singular is caviola, so take off the E, so it's not caveolus, it's caviola. So what are caveolae? What are these little caves that we're talking about? Well, they are permanent invaginations of the cell membrane, and we believe that most cells do actually have these. So, just like the cytoskeleton is a complication inside the cytoplasm, caveolae are a complication of the cell membrane. So, once upon a time, and throughout this video actually, we like to draw cells like this. This is the plasma membrane picture that we like. A nice, smooth cell membrane like so. However, for most cells, this is not the case. Firstly, as we've just discussed, often you'll have clathrin-mediated endocytosis occurring, which will cause disturbances to the cell membrane. But more than that, you have these permanent little pits called caveolae present in the cell membrane. So the actual picture of this looks more like this. You have these little pits in the cell membrane all over the place. And these are what are called caviolae, and I've got bored of drawing them now, so I'll just continue it on like that. You have loads of these, so in reality I should have done it all the way around. You have these little caves, these little invaginations of the cell membrane. These are the caviolae, singular caviola. So this is a caviola, we've got caviolae on the cell membrane. So, let me tell you a little bit more about these special portions of the cell membrane. So the first thing to say is that the actual lipid in these portions of membrane is not actually the same as the portions of membrane that are flat. So if you look at the lipid in these portions and compare it to the lipids in these portions, the caviolae, it's not actually the same. The caviolae have weird lipids in them. So let's firstly discuss this. So, one of the lipids that they contain a lot of is cholesterol. So, cholesterol is a normal component of the cell membrane. Of course, I should probably just remind you about normal components of cell membrane. So let's talk about normal components of cell membrane. So the major component of cell membrane is phospholipids. Phospholipids that are majorly phosphatidylcholine. So if I go back to my picture, where is it? Of phosphatidylinositol 4,5-bisphosphate up here. Remember, this is the structure of phosphatidyl. It's the basic structure of phospholipid. Glycerol with 2 long-chain carboxylic acids, the lipid molecules there, and a phosphate group here, and that's called phosphatidyl. Now, the major phospholipid that you have in the cell membrane is something called phosphatidylcholine. So I'll just put this here. So instead of having phosphatidylinositol, we have phosphatidylcholine. And in phosphatidylcholine, instead of having inositol there with 4,5-bisphosphate on, you instead have a molecule called choline. And this is the same molecule that is used to make the famous neurotransmitter acetylcholine. And this molecule, phosphatidylcholine, often just abbreviated to PC, has another name. It's also called lecithin, and it is, as I say, the major component of the cell membrane. So that's one of the major things that you have in a normal cell membrane. So let's just come down here. So if I just draw a little bit of cell membrane here, so in white here, this is my cell membrane. Often we just draw phospholipids with this very basic picture where we have a head, like so, and then 2 lipid tails, and then if we draw the inner leaflet as well, there is another phospholipid, here's another phospholipid. These will mainly be phosphatidylcholine molecules, PC molecules, and this head is representing the phosphate group and the choline molecule that's attached onto the phosphate group, whereas the 2 tails are representing the 2 long-chain carboxylic acids attached to the first 2 alcohol groups of the glycerol molecule, which is the backbone for the entire phospholipid structure. So, that's the major component of cell membranes. In addition, you do have cholesterol molecules. Now, cholesterol molecules are very hydrophobic, except for one glimpse of polarity. They do have an alcohol group. So if I want to draw a basic picture of a cholesterol molecule, I might draw it like so. So this green line is representing a cholesterol molecule. Most of it is very hydrophobic and therefore needs to be in the core of the phospholipid bilayer with these long-chain carboxylic acids, but then it has one glimpse of polarity up at the top, which is this red bit, which is the alcohol group, and that faces out just like the phosphate groups are polar to interact with water molecules. So you can have cholesterol molecules in the outer leaflet, in that case the alcohol group will be facing out to the extracellular fluid, all cholesterol molecules in the inner leaflet facing in towards the cytoplasm. Well, the alcohol group would be facing in towards the cytoplasm, and the hydrophobic rest of the molecule would be in the core. So cholesterol is a normal component of the phospholipid bilayer. However, in these caviolae, you have loads of cholesterol, much more cholesterol than you would normally have. So this is the first strange thing about caviolae, lots of cholesterol. It's normally present in the phospholipid bilayer, but not to the extent that it is present within the caviolae. So lots of cholesterol in the caviolae. The other thing, which I'm going to go into more detail of, I'm not drawing out the actual structure of cholesterol because, you know, everyone's familiar with what cholesterol is. You'll have heard of it before, so I don't feel the need to stress that. But I'm now going to go into the detail of this second component in more detail because people are generally less familiar with this. The other strange type of lipid that you have present in high amounts in the caviolae are what are called sphingolipids. So, what is meant by a sphingolipid? So, a sphingolipid is derived fundamentally from a molecule called sphingosine, and we will start, therefore, by discussing what is the structure of this molecule. It's called sphingosine, and we'll build on from there to get to what a sphingolipid is. So, it's helpful actually to look at the proper name for sphingosine. Sphingosine is kind of the biochemist's name. Of course, chemists have a name for sphingosine, which is much longer, but which is more useful because it tells you about the structure. So, let me hope that I can get this right. So I think the name for sphingosine is 2-amino and then 4-octadecene. So octadecene, and that wasn't brilliant planning. I've already run out of space. And then it should be linked up 4-octadecene or 1,3-diol. Okay, so sphingosine is 2-amino, 4-octadecene, 1,3-diol. That sounds good to me. So octadecene, let's start with this. This means that we're going to have an 18-carbon molecule. So I'm going to start by drawing the carbon backbone here, and we'll do it in skeletal structure, of course. So let me pick another colour, red. So don't lose count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. Whoops, what am I doing? Not 19. Oh, no. Now I'm going to have to do the entire thing again. Actually, no, I can redo it and just cancel out the last one. Sorry, I got a little bit carried away there. Let me just confirm that I have drawn over too many. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Yes, we need to get rid of that final one. Where is the... Oh no, I've lost the rubber tool now. We will have to redraw it. I'm sorry about this. Excuse this moment of unprofessionalism. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. There we go. We now have the correct carbon backbone of 18 carbons. Now all we need to do is add a few funny groups in occasionally. So let's start off with this bit actually. 4-octadecene, the ene. If it was octadecane, that's what I've drawn at the moment. Octadecane, a 6-carbon fully saturated hydrocarbon here. Sorry, an 18-carbon fully saturated hydrocarbon here. But I don't want it to be fully saturated. This is ene, so we're going to have a double bond at some point, and that double bond is going to be coming off the 4th carbon. It's going to be between the 4th and the 5th carbon. So I think it will be in this position here. So 1, 2, 3, 4, and then between the 4th and the 5th carbon you have a double bond. That's 4-octadecene. What I've now drawn is 4-octadecene. Now it should be easy. We just have to stick an amino group on 2 and 2 alcohol groups on 1 and 3. So I hope I can't go wrong with this. So off 2, over here is the amino group, NH2. So we've taken the hydrogen off and put the amino group on. And now an alcohol group comes off 1. So here is the alcohol group coming off 1 there. Just try and connect it up. And then an alcohol group is also going to come off 3, like so. So just circling a few things. These alcohol groups, this alcohol group here, and this alcohol group here, those are the 1,3-diol of this name. The 4-ene bit is this double bond here. And then finally, the amino group, the 2-amino here, is this bit of the structure. Okay, so this is the molecule that we call sphingosine, and this is the basis for creating sphingolipids. So, before we say what a sphingolipid is, we need to talk about what a ceramide is, because sphingolipids are modified ceramides. So next piece of terminology is, what is a ceramide? A ceramide is a molecule derived from sphingosine by taking a long-chain carboxylic acid, and this doesn't need to be a specific long-chain carboxylic acid, you just take some long-chain carboxylic acid, and biochemists like to call long-chain carboxylic acids fatty acids. So you take a long-chain carboxylic acid or a fatty acid, and you bind it to this amino group via an amide link. So trying to draw a little bit of this on, in fact I won't try and draw this on, instead I'll draw a little separate picture down here. So if this is the amino group on that second carbon here, you'll take one of the hydrogens off, and then you're going to have an amide link to a long chain carboxylic acid here. So there's the carboxylic acid group bound via amide link to the nitrogen of the amino group, and here's its R group, which is not set in stone. You can put multiple different fatty acids there, and therefore there are multiple different ceramides. So it's not as though there is just one molecule that is called the ceramide. There are loads of ceramides. So I'll put an S on there. So, from sphingosine then, you can get ceramides. Now, let me teach you a little abbreviated drawing that we can make for a ceramide. So this drawing is going to be in the same spirit of that drawing that we did for phosphatidylinositol 4,5-bisphosphate. You see, this great big tail of the sphingosine molecule, this is effectively just like a fatty acid. It's very, very hydrophobic. This bit here, where we've got alcohol group, and amino group, this is similar to, effectively, glycerol in the phosphatidylinositol molecule. So it's as though we've got a backbone here, a long-chain carboxylic acid, we're sticking another long-chain carboxylic acid on here. So the molecule actually ends up being very similar to a phospholipid molecule. So I could draw a little abbreviated drawing here. This is the sphingosine backbone, and then we've got another long-chain carboxylic acid that we've attached on there via this amide link onto the amino group, and this is my abbreviated drawing for a ceramide. And of course you're going to have alcohol group here as well, making this head polar. So you've got a polar head with two tails, so you could use a very similar drawing to this. In fact, you could use the same drawing for this ceramide molecule. Finally, to create a sphingolipid from a ceramide molecule, all you do is stick something else onto this head. You stick something onto the alcohol group on carbon number 1 here. So you can stick all sorts of moieties, all sorts of different molecules on here, and they're all called sphingolipids. So to turn the ceramide into a sphingolipid, you stick something on here. So I'll just call this a moiety of some sort, chemical moiety. And now this molecule is a sphingolipid. So this is what sphingolipids are. You take sphingosine, you bind another fatty acid to it, you've then got a ceramide, modify the head to make it more interesting, you've then got a sphingolipid, and these can sit in the plasma membrane just as you would expect, the way that a phospholipid would, with the long chain hydrophobic tails here pointing into the centre, and the head, which will probably be hydrophilic, able to interact with water, polar, it will be facing out either to an extracellular fluid or to the cytoplasm. Now, these types of lipids are very heavily present in the caviolae. Again, you'll have a few of them in the normal plasma membrane, but they're hugely enriched in the caviolae. So caviolae have these weird combinations of lipids. They have very high cholesterol level and very high sphingolipid level. So let me just put an arrow there. So sphingolipids are going to be high as well. So the next question is, what makes caviolae bend in like this, because it is not just the weird combination of lipids that you have in these regions that makes the membrane bend in. You have some proteins in this membrane as well, along with the funny lipids, and those proteins are responsible for the membrane being invaginated in. So caveolins is the next topic, which are these funny proteins. So we'll come down here and we'll go to having a blue pen. So next topic are these proteins called caveolins. So caveolins are proteins associated with caveolae, hence why they were named. So the caveolae were discovered first, named because they were little caves in the cell membrane, and then caveolins were proteins discovered to be associated with the caveolae, hence why they were called caveolins. So there are multiple different caveolins, 3 in fact at present that are known, and they're quite simply called caveolin 1, caveolin 2, and caveolin 3. And for short, we abbreviate caveolin down to CAV like so. So we've got cav1, cav2, and cav3. And they all have the same membrane spanning topology and the same function in creating caveolins in the cell membrane. They have slightly different tissue distributions. So different tissues will use different caveolins. But they all do pretty much the same thing. So let me now discuss the structure of a caveolin. So it has a very weird membrane spanning topology. It doesn't span the membrane fully, although it does span it to some degree. So let me now draw this in here. So again, let's have a bit of cell membrane here, like so. And now let's draw a caveolin in this cell membrane. So they are proteins that are on the cytoplasmic aspect of the cell. So this is going to be the extracellular fluid side, and this is going to be the intracellular fluid side. So these caveolin proteins, here is going to be the amino terminus of the caveolin protein. Then it has a portion that is going to go into the cell membrane, but it's not going to go all the way through. It's going to go through into the middle, i.e. between the inner and outer leaflets, and then it's going to come back out like so, and here is going to be its C-terminus. So this is the membrane spanning topology of all of these caveolin proteins. So this is a caveolin protein, whether we're talking about caveolin 1, caveolin 2, and caveolin 3. Now, all of the caveolins have oligomerization domains that are in this position here. So that domain I've now boxed in red, this is an oligomerization domain. So they're going to oligomerize, which means they're going to join with other caveolin proteins to form an oligomer. An oligomer is a multi-subunit structure. It's like a polymer, but not as big as a polymer. So polymer means that you've joined loads of a subunit together to make the whole thing. Oligomer means that you've joined some. It implies not as many things have been joined together as you would if you were using the word polymer. So this is an oligomerization domain. And now what's going to happen is that caveolin proteins are going to oligomerize, and they oligomerize together into about sets of 14 to 16. So 14 to 16 of them are going to join together. Now it's quite difficult to actually show this convincingly, because really again you need to be able to draw a three-dimensional picture, trying to find that same sort of yellowy colour back again. So here is another caveolin protein. So really in my drawing I can only show 2 of them joining together here. So here's 2 of them, but you have to imagine this is a 2-dimensional picture here. You can imagine that you could have another one which is oriented in the perpendicular plane. In fact, I'm going to try and draw it from above. Let's draw it from above. So imagine we're now standing above and looking at this caveolin oligomer. So here is the cell membrane viewed from above, and now if we're drawing caveolin proteins, here are the 2 that we've drawn so far viewed from above. So we're now only seeing them, a little bit of them, with seeing them from this aspect. So here are the 2 caveolin proteins. They're joined together with their oligomerization domain. So I'll get the red pen to colour in the oligomerization domain in the centre there. But you won't just have those 2, you'll also have other ones coming out in different planes. like this, and overall you have around 14 to 16 of these joined together, so it makes a sort of little umbrella shape, I suppose. Here's like the stem of the umbrella, and all of these will form like the roof of the umbrella together. So 14 to 16 caveolin proteins join together, they oligomerize together to make one of these caveolin oligomers, and these are present in the cell membrane of the of the caveolae, and these are what are going to bend the membrane inwards. And in addition to oligomerizing into these caveolin oligomers, separate caveolin oligomers will join together via their C-terminals. So let me now draw a picture of a caveola in detail. So I'll come over here, and I personally think that the word caveolus would be far better for single caveolae. However, I will force myself to use the word caveola. So here we go. Here is the cell membrane in green here. Here is our pit that's going to be the caveola, and here is the inner leaflet, like so. And we know, of course, that the lipids of this portion of membrane will be different. We've been through that now in detail. Lots of cholesterol and lots of sphingolipids. Now let's draw some of the caveolin oligomers in this portion of membrane joined together. So here we go. Here is one caveolin oligomer. There's one caveolin protein of the caveolin oligomer at the moment. And there's another one. Again, because I'm drawing a two-dimensional picture, it's difficult to add more in. I think it will ruin the picture if I try to draw some coming out of the page towards us. Okay, now let's draw another caveolin oligomer. So here's another one, like so, and so on. We can have another one here. And they're all linked together by their C-terminals, so these portions can connect together. So different caveolin oligomers will join together so that this entire inner leaflet of the caveola is lined by caveolin oligomers. And these are what are actually bending that membrane inwards and maintaining this caveola. So let me label it up. So this is a caveola. So I hope we now understand what caveolae are. These are permanent structures that are always there, and in most cell types in the human body you do have these. So the next question is caveolin-mediated endocytosis. These things can be used for endocytosis, and it's easy to understand how. We have these formed invaginations of the cell membrane. All we need to do is clip off the top, and then we have a caveolin covered vesicle that can then go into the endocytic pathway. So it's easy to understand how these things can be used for endocytosis. Now the trigger pathway isn't well understood, so we don't really understand what it is that suddenly decides that a caveola is no longer going to remain permanently on the cell membrane, but is instead going to be cut off and endocytosed into a vesicle. We don't understand that trigger. Also, what we need to say is that we don't really understand how proteins receptors that might be cargo laden, they might have cargo bound to them, are going to be recruited to the caveolae. What must happen, however, is that if the virus particle bound to the secondary receptor is going to be endocytosed by caveolin-mediated endocytosis, then the secondary receptor with the virus particle must somehow get into caveolae that are bound for caveolin-mediated endocytosis, or potentially it will be the thing that actually triggers the caveola to be endocytosed. So, doing this in chronological order, the virus will be bound to the secondary receptor. Maybe then the secondary receptor will have a change in conformation of its intracellular aspect, and it will be recruited maybe to caveolae. We do know that loads of different receptors do localize to caveolae, so another possibility is that actually the secondary receptor might be present in caveolae all the time, and then potentially when the virus binds to the secondary receptor, it triggers a change in conformation on the intracellular aspect that might lead then to the caveolus being endocytosed. We don't understand the trigger mechanism. However, we do know a little bit more about how it actually occurs. It occurs again through dynamin. So what you do is you just, again, assemble dynamin oligomers around the neck of the caviola, and that will form a spiral. And again, the dynamin molecules, they are GTPases, they all bind GTP and then synchronously will hydrolyze that GTP to release energy, causing them to constrict around the neck of the caviola and cleave it off from the cell membrane. So it's again via dynamin that caviola can cleave off from the cell membrane, just like what we've got drawn out here, except this time, instead of the pit here being coated by clathrin, it's coated by caveolins instead. And then what you have is an endocytic vesicle that's coated with caveolins. This can then go to the early endosome, just as an endocytic vesicle from the clathrin-mediated endocytosis process would go to the early endosome. So that is what we know at present about caveolin-mediated endocytosis. So let me summarise what we've just been through, because it's a complicated story, mainly because so much is unknown. The clathrin-mediated endocytosis process is much better understood than the caveolin-mediated endocytosis pathway. So let me highlight what we do and what we don't know. So what we know is that caveolae are present in many different cell types. They'll be present in the basal cells of the squamous stratified epithelium that our HPV virus is going to infect. We know that they have funny lipid composition and that they are maintained, these invaginations are maintained by many caveolin oligomers in the inner leaflet of the phospholipid bilayer of the caveolae. For reasons that we don't understand, some caveolae can then undergo an endocytosis process. They can be cleaved off in a dynamin dependent fashion. So again, dynamin will oligomerize around the stalk here, and then synchronously the dynamin monomers of the oligomer will hydrolyze GTP, and the whole spiral will constrict to cleave the membrane of the caviola off from the plasma membrane, and then you'll have a caveolin-coated vesicle. We don't know, as well as we do for clathrin-coated vesicles, whether the caveolins have to be removed from the inner leaflet of the vesicle before it can then be join the endosome. Potentially they don't, though. So the caveolin-coated vesicle will then go to the early endosome, and of course, if the virus is inside that caveolin-coated vesicle, it will be ending up in the early endosome. So we don't understand the trigger. We also don't understand as well how cargo receptor complexes localize to caveolae that are going to be endocytosed, and of course that goes along with us not knowing much about what it is that actually triggers the endocytosis of a caveola. But overall, if the virus is going to use caveolin-mediated endocytosis to get into the endocytic pathway, the secondary receptor with the virus bound to it must either already be in the caveolae, or it must localize to the caveolae once the virus has bound to it, or once the caveolae decides that it wants to be endocytosed, and then it will end up in the early endosome, just as it would if it was going through the clathrin-mediated endocytosis pathway. Okay, so we'll have a break here. In the next video, we'll discuss macropinocytosis.